All right, I think we'll get underway. Let me just switch on my mic here. So I'm Jordan Smoller, and I'm part of the Stanley Center here at the Broad, and also the Center for Human Genetic Research at Mass General. And I'm going to be talking with you a little bit about the question of the importance of phenotype, which I think is a fascinating issue that we probably don't pay as much attention to as we maybe need to. And this is a series in which you have heard some of the startling advances in our abilities to assay the genome, to look at every single base in the human genome, to look at structural variation, to look at mutations and common variants and so on. But if you're doing a genetic study at some level, you're trying to connect genotype to phenotype. And the phenotype part of the equation can sometimes be the most challenging, maybe the rate-limiting step in some cases. So I want to talk with you about some of the issues that we need to consider in thinking about phenotypes. And my subtitle there is with Tales from Psychiatry, because psychiatry is an interesting field with respect to this, in that our phenotypes are a moving target, maybe more than many fields. And as I sometimes say, if you can make it there, you can make it anywhere. It's sort of the New York of phenotypes. So let's start with the fundamental question: what is a phenotype? Does anybody know this guy, Wilhelm Johansen? That name. He is the guy. He was a Danish botanist, and he actually came up with the term phenotype. He also came up with the word gene. So this is a guy who was good at branding, and sadly, though, maybe not so good at branding, because he's not as well known as maybe he ought to be for those kinds of contributions. Anyway, the general definition that we use, and this is pretty general, of a phenotype is the observable properties of an organism. So that pretty much covers everything. It also highlights, I think, that our ability to see phenotypes is in part our ability to measure them, and technologic advances can create new phenotypes for us to study, as they often do. So I want to talk a little bit, dive into this area with you and address a few issues. One of them is, how do you select a phenotype for a genetic study, for example? If you're trying to identify genetic variants that influence something, how do you decide what that something is? How do you best then measure it? How do you decide how you're going to assay that phenotype? And as we'll see, there are a number of issues there. And then, are there ways in which defining phenotypes can get you further in terms of gene discovery, or maybe actually help you characterize the way that genes are acting in unexpected ways? So we'll touch on some of those issues. And many of you may be not so immersed in the world of actually measuring phenotypes, but we care about this for a number of reasons, no matter what we're doing in genetic studies. First of all, if we're interested in finding genes, we have to have a phenotype in mind, a phenotype that we're actually looking at. And it turns out that that's not often a very straightforward thing. But if we are not so precise about how we measure our phenotypes, we can actually lose a lot of power. It can make a big difference to our ability to identify genotype-phenotype relationships. And then I'll talk a little bit at the end about how you can use phenotypes not only for discovery, but for characterization on the back end when you've identified genetic components of a phenotype. You may begin to wonder, what is the phenotypic spectrum of those genetic components? How do they play out at the level of biology and other observables that we might now be in a position to look at? So I'm going to start off with where we're coming from in psychiatry. And as many of you may know, in psychiatry, we're actually, in some ways, very lucky, right? Because if we wanted to find phenotypes, all we have to do is go to the bookshelf. This is the book, the DSM. everybody familiar with the DSM? You've heard of this, right? The Diagnostic and Statistical Manual of Mental Disorders. This is a book of phenotypes. If you go into this book and you peruse it, what you will see is a list of diagnostic criteria for everything that psychiatry essentially officially recognizes. Because when you look at those criteria, you notice that there are some decisions that were made in defining these phenotypes. For example, if you want to define major depressive disorder, depression, you'll find that you need, according to the book, 2 weeks of a pervasively depressed mood or loss of interest in things, and then you need 4 out of 9 of a list of things to have occurred at the same time, and you need it not to be related to some other medical disorder and so on. But you can see immediately there are some arbitrary seeming decisions made there. Why 4 out of 9? Why 2 weeks? And the answer is that a consensus of experts got together and said that's a pretty good definition. And it turns out that an enormous weight of things are hanging on that. Clinically, for example, we use those definitions to decide who needs treatment. Insurance companies use those definitions to decide what they're going to pay for. And in genetic studies, we tend to use those definitions because they are the canonical definitions at this point. But it's important to recognize that they are humanly defined syndromes. And actually, if you look at the history of this book, the DSM, which actually started in 1952, this is the number of diagnostic labels in the book. So over the past 60 years, we've seen this incredible growth in the number of sort of phenotypic categories. And that makes you worry a little bit about how stable these things are. So let's talk about what makes a good phenotype. This is one of my favorite examples of an innovative phenotype. You know when you're on the phone with somebody and they say, I'll let you go now, like they're doing you some kind of favor, but it's really them that wants to get off the phone and they just don't want to admit it. I think I've isolated the gene for that. So, okay, maybe that's not your ideal phenotype. On the other hand, we do see in the literature papers about interesting phenotypes. Here's one about gene variations associated with creative dance. And there's actually a biological kind of model for how that might work. But a paper on the relationship of a dopamine receptor to skiing and snowboarding behavior. And one of my personal recent favorites is polymorphisms in the oxytocin receptor gene. I actually explain why sales professionals love to help customers. Okay, so is that a reasonable phenotype? Well, I guess it was reasonable enough to do this work. But when we're thinking about what's a good phenotype, we have to ask a few basic questions, right? We're asking, is some version of this phenotype that we're interested in influenced by genes? And that is, even before we identify perhaps the specific genetic components, we want to know, is there evidence to think that we're going to be able to find genes related to this, if that's our goal? Our goal is to, say, do an association study or to identify mutations that may have large effects on some phenotype of interest. Is there reason to think that there are going to be such things out there? And if so, what's the best way to define that phenotype in a way that is going to maximize the genetic effects that we can find? Because you could imagine that if I asked you to do a study of, or if you were interested in heart disease, and you decided that, you know, I want to do something that, let's imagine it's 100 years ago or so, and you're interested in heart attack, but you don't really have great ways of measuring when an actual heart attack has occurred. You don't have cardiac enzymes, you don't have cardiac imaging that we now have. And so you kind of go on chest pain and maybe an EKG, although frankly the EKG still is pretty useful. That might not be as powerful as if you were able to further dissect that phenotype in ways that, for example, we may be able to do now. So you want to find what's the best definition. And then another thing you want to know about your phenotype going in is, how complex is the genetic architecture? By genetic architecture, we mean, of course, the number and effect sizes of the and frequencies of the genetic components involved. That turns out to be obviously very important to your success in identifying genetic components, but it is something that is very difficult to know in advance. We'll talk a little bit about that. Okay, so the first thing you might want to know is there some reason to believe that genes are going to be underlying this phenotype? And the first question that people sometimes ask, and this was a question you could ask well before the genome was, sequenced, or we were doing the kinds of genetic studies we're doing now, and that question is, does it run in families? If it doesn't run in families at all, it doesn't seem to, then you might be a little bit less enthusiastic about pursuing this. So to answer that question, we can do what's called a family study of our phenotype, and this is basically a very simple case control study. So we're comparing the prevalence or risk in relatives of affected individuals to the prevalence in relatives of unaffected individuals. If it's more common in the relatives of affected individuals, that suggests it's aggregating in families. And we can actually get a kind of index of this, which is the recurrence risk ratio, which is defined as the ratio of the risk to relatives of type R, so let's say first-degree relatives, compared to the population risk. So how much more likely are you to be affected with this phenotype if you are, say, the brother or sister of somebody with the phenotype, compared to if we selected you at random from the general population? If that is significantly greater than 1, it suggests that this is something that's running in families. And here's, for example, the sibling recurrence risk ratio, just spelled out in different words. Now, it turns out that a lot of things run in families. And in fact, there's quite a lot of variability in terms of the recurrence risk ratios for many things. I've just listed a few here, heavy on the psychiatry, because as I mentioned, that's an area of great interest. But here's a sampling. So you've got autism at about 50, multiple sclerosis at about 20, early onset MI at about 3, breast cancer at about 2. Maybe these aren't the numbers you would have necessarily predicted, because some of these have been easier, more tractable in genetic studies than others. There are a couple of things to notice about this. One is that there's a big range, and that range might be a little bit misleading. There can be biases that inflate these numbers, but even putting that aside, so for example, you might be biased in how you collect your family study sample, or people might over-report if they've got a family history compared to people in the general population for whom this is not something on their radar all the time. But more relevant, I think, is that this is kind of a constrained number, because remember, it's a ratio. So if the population prevalence of your phenotype is common, then this number can't be that high, right? If you've got a disorder that's affecting 20% of the population, 5-fold increased risk is about your upper limit. And the other thing, of course, is that these family studies are limited in the sense that we inherit not only our genes, but our environments in our families. These are other things. So things can run in families for reasons that are not necessarily genetic or heritable. So the next thing that people often want to know is this thing actually heritable? And before the genomic age, our ability to answer that question was largely limited to sort of experiments of nature, like a twin study. Because, of course, there are two kinds of twins, identical monozygotic twins and non-identical dizygotic twins. And the identical twins are, for all practical purposes, clones. So you've now got a pair of people who are genetically identical and a pair of people who are only half genetically similar. Now let's assume that the environment is treating them the same. To the extent that the identical twin pairs are more similar to each other, that suggests that genetic influences are playing a role. And if the identical twins are not completely similar with respect to your phenotype, which pretty much they never are, that implies that there's something beyond genetic variation contributing. And here we get this index, this number, the heritability, which is essentially the proportion of phenotypic variation or variance in a population that's attributable to genetic variation. This is a number that, and a term that we use all the time. It's a little bit of a tricky one for reasons that I will mention a little bit more, and often misunderstood, and often maybe thought to carry more information than it does. But basically what it's saying is that in the population, how much of the distribution of risk, distribution of cases, variation, is attributable to individual genetic differences. People talk about two kinds of heritability for a phenotype. One of them is narrow sense, which is the one we typically refer to, which is the additive effects of genetic variations. And there's broad sense, which includes not only additive effects, but dominance effects and gene-gene interactions and so on. These numbers don't account for gene-environment correlation, meaning that there are cases, for example, in psychiatry, where there is a genetic influence on whether you're actually in an environment that may create the phenotype. So for example, in post-traumatic stress disorder, it turns out there's a heritable component to whether people are exposed to trauma, which can increase their risk for post-traumatic stress disorder. Those kinds of things may inflate the apparent heritability. Gene-environment interaction, environments that modify, in some groups raise the risk, and in other groups lower the risk, are not really accounted for. Epigenetic effects, which may be making twin pairs more similar, may also be playing a role. So there's some imprecision there. Once again, the heritabilities vary quite a bit. I've loaded this up with psychiatry, but here are some heritabilities. And of course, heritability ranges from 0, no impact of genetic variation, to 100%. It's never either of those numbers, it's always in between. But what you can see is that for the psychiatric disorders, for example, these numbers are pretty high. Interestingly, for something like breast cancer, where identifying specific genes came relatively early, speaking, in the 1990s, the overall heritability is low. How is that possible that you've got a phenotype like breast cancer where there are major genes like BRCA1 and BRCA2 where mutations are relatively highly penetrant, and yet its heritability is lower than something like ADHD, which has a very high heritability, but has been very difficult to identify genes involved in. What does that tell us? It tells us that the heritability is not necessarily a good index of that question. And the reason is that, remember, this is a population measure. So this is kind of the total picture of genetic influences. And while you might have a mutation that tracks and is highly penetrant in a family, if it's not contributing to a great degree overall to phenotypic variation, its contribution to the heritability may not be that great, and the overall heritability might be actually not that great. It is now possible to move beyond what we used to do in terms of estimating these things from twin studies to actually estimate them from genomic studies. And one of the ways that this is now increasingly done is to look at the heritability of genomic data from VWAS studies, for example, sometimes called SNP chip heritability. And we can estimate this, and I'll refer you to a really nice, very easy to understand review from Naomi Ray in the Journal of Child Psychology and Psychiatry, but there are a number of reviews that she and others have written about this are very nicely lay this out. Essentially what you're doing is taking unrelated individuals and looking at the relationship between genetic similarity in your sample to phenotypic similarity. The principle is not that unlike a twin study, except in the twin study you had two types of genetic relationship. Now you've got a lot more types of genetic relationship. But you can now, with genome-wide data measured across the genome, create a matrix of genetic similarity, and you've got your matrix essentially of phenotypic similarity, and you're looking at the relationship of those two. And using a residual maximum likelihood analysis and linear mixed modeling, you can pull out variance components, including the heritability here. And this is really useful because it gives us a sense of not only in principle what the heritability is, but actually using genomic data, is there likely to be a signal here? An important caveat is that this heritability is based on common variants, so it's not going to be the entire heritability, even the entire additive heritability. It's not including most rare variants. And so for that reason, the numbers tend to be a little bit lower. But you could use this, as we do in using twin studies, to use the heritability to kind of prioritize what are the phenotypes that you think are maybe most valuable to go after. And sometimes you actually have a vast number of phenotypes. This is a paper that'll be out next week, actually, with a method to do this kind of analysis, even in the case where you have millions, perhaps, of phenotypes that you're trying to sort through and prioritize using a different method, kernel machine regression-based method. And for example, in neuroimaging, where you've got many, many, many possible phenotype definitions, having a very high throughput kind of metric that can sort through those may be extremely helpful. We did this with a large brain imaging sample, and the difference when you get up to massive amounts of possible phenotypes, as you might also see in electronic health record registries and these kinds of things where we've just got so many phenotypes that we might want to explore, the difference between traditional methods and newer methods can be substantial in terms of just computational time and burden. Yeah.
How do you deal with multiple testing in that scenario? I mean, you can explore millions of phenotypes, and you're going to always find many that seem to look inflated for heritability.
Yeah, right. Well, you have to account for that. In this case, this is a method that can do actual permutation testing. So it will account for it and give you a permuted empirical p-value. But you're absolutely right. This is a big issue when you're looking at phenotypes in general. When the more you test things, the more you're going to, especially if you're doing hypothesis testing of phenotypes, your type 1 error rate is an important thing to remember to control. You still might, though, be interested in the rank ordering of effect sizes, which, you could get a sense of here, but absolutely right. Here's the flip side, though, just to remember. This relates to what we said before about what heritability can and can't help us predict. So here are two, is it true that the more heritable, the better in terms of selecting a phenotype? Here's some pretty heritable phenotypes, right? Height, about 90%, weight or BMI, about 70%. Does anybody know what this is a picture of? So I'll read you the names here. Sean Connery, George Lazenby, Roger Moore, you're probably guessing at this point. These are, of course, the various James Bonds. And you'll notice quite
That's not to scale.
Well, it's not.
5'8"
Yeah, okay, it's not to scale. It's drawing out a point. But yes, the point is there was phenotypic variance in the casting of James Bond. I think that's the important takeaway today. No, but here's the point. So highly heritable. Does that mean it's going to be easier to find genetic components? Well, not necessarily. In the GIANT consortium, these were some of the early results at this point, where people were able to put together these phenotypes on massive samples, like 250,000 individuals or 180,000 individuals. And you could identify, for example, for BMI, 32 genome-wide significant loci with 250,000 people, explaining about 2.5% of the variance. So the heritability by itself is not answering the question of how easy this is going to be. You certainly want a phenotype that is heritable, but it doesn't tell you much about the genetic architecture. And in some ways, this is sort of the ideal case. You've got something that's highly heritable, easily measured, precisely measured. It's pretty easy to measure weight. So why would it be so difficult? And the answer, undoubtedly, is there are lots of reasons you could expect these would be particularly complex phenotypes, right? Because practically everything, they are the integration of so many different mechanisms in the body. How tall you end up being is a function of all kinds of metabolic processes, bone development, hormonal and other things. And so maybe not surprising that it's going to be a relatively complex phenotype. Just a couple of things to again remember. The heritability means that if you had a heritability of 60%, that means at least some gene or genetic variation is operating, and that 60% of the individual differences in a population are attributable to genetic differences, additive genetic differences. It doesn't mean that 60% of your obesity or something is genetic and 40% is environmental, you as a person. It also doesn't mean that environmental interventions cannot have a significant effect on this phenotype. It's really not telling you that. And it doesn't say much about genetic architecture, as we just talked about. And it depends on the degree of genetic and environmental variability in the population, because this is a variance component. It is the ratio of genetic variation to overall phenotypic variation. And the overall phenotypic variation is a function of not only genetic variation, but environmental variation and noise and so on. And this is, twin studies have to some extent given some nice demonstrations of this. This is a study of the heritability of IQ, in which the additive genetic component, this is the heritability, the common environmental impact, and the individual specific environmental components were separated out, and this is socioeconomic status. And what this study suggested was that the heritability of IQ varied over socioeconomic status. That's weird. Why would that be? Whereas environmental factors, environmental variance, seem to play a bigger role there. Anybody imagine an explanation for that? Well, one explanation that's been given is that when you're in a situation of deprivation and resources might be scarce and small genetic differences are not going to matter that much. What's going to matter is to the trait variance, much more is environmental effects. When you've reached a sort of point where those environmental effects are essentially, the environmental deprivation is sort of taken care of, then you can see some of these genetic effects show their effect, essentially. So it's an interesting illustration of that. And this makes the point, again, that the genetic architecture of your phenotype is not well revealed by its heritability. So here's a case where you've got two situations, a rare variant situation and a common variant situation. So if you look at a Mendelian dominant phenotype, you could end up with a locus that contributes 12% of the phenotypic variation. That is, its heritability is 12%. If it were, say, a dominant mutation that was very rare, but that had a huge effect on the phenotype, or for a more common disease, a smaller effect on the phenotype, but a more common variant. These both add up to the same contribution to heritability, which tells you that just knowing that heritability number doesn't tell you, this might be a much easier scenario for identifying a disease-causing locus. Okay, so let's say we have the necessary but not sufficient condition that we've got a phenotype that's familial, heritable, and now let's turn to the idea of measuring phenotypes and what our considerations are. The first one is sort of,
Jordan,
yeah.
before you do that, just to go back, that's a nice figure, this last one.
Credit Neil Risch, yeah.
Yeah,
Yeah.
it kind of presents it as an either or. And in fact, some of these are ands. In other words, the Mendelian phenotype that you find out there, type 2 hypercholesterolemia, a variety of MODY genes that are out there by themselves in the Mendelian context, actually happen to be also in the common context, just not exactly the same mutation. They're common variants in it.
Yeah.
So this kind of presents it a little more dichotomously than it really is. In fact, we're finding that there's a blend of these.
Yeah.
Correct?
No, that's true. That's true. And this is really just to illustrate the point that if you had these two scenarios, you could end up with the same thing. But you're absolutely right that any given locus can be both contributing to overall heritability as a rare, highly penetrant. There might be other variants in that same locus that fit much better with the bottom picture. So some basic things that I don't think will be controversial. If you don't measure the phenotype well, that's going to be a problem. And you're going to have imprecision and misclassification that could make a big difference to your power. And a question related to this is, how should you model the phenotype? Is it a quantitative trait, or is it a diagnostic category, or a qualitative distinction? And of course, that has big implications for how you analyze the data. Are you going to do it as a linear kind of effect, or as a case control, or logistic regression, or kind of analysis? And it depends on what the state of nature is, which we typically don't really know. But you can imagine that there will be scenarios where if your trait is in fact dimensionally quantitatively distributed, and you model it as some cut point that you arbitrarily say you're above it or below it, that's going to be a problem in some circumstances. So let's say something like blood pressure. Hypertension in the medical community is traditionally defined as 140 over 90, either 140 or above or 90 or above on the bottom, the diastolic. And, does that mean that there's a qualitative difference between this person here and this person here? No, of course not. But if you throw them into two different piles, you're kind of losing that information. Now, if in fact you've sampled the extremes and you've gotten a case control sample, then measuring the sort of quantitative variation is not going to make that much of a difference, because you've essentially divided your sample into two ends of the distribution. So, this is what you've got. You've essentially divided. And there are circumstances where case control actually does better, that is dichotomizing. And it depends in part on what the underlying prevalence of the disease is. But for diseases where the prevalence is less than, say, 10%, so not very common diseases, having a case control design can sometimes boost your power, because you're essentially enriching the number of cases. And through simulation, at least, you can see that this ends up giving you greater power as you move up in your proportion of cases for a quantitative trait. But if your disease prevalence, that is, if the cut point is less than 10%, sorry, you actually may do better with the case control design. How you model it can make a difference to what you find. This is not necessarily the most dramatic example of that, but here's a simple case where you do a study of nicotine, cigarette smoking, and you could model that as cigarettes per day. That's a common phenotypic measure of this, or a dichotomous variable of nicotine dependent or not, which is actually a psychiatric diagnosis in the book. And whether you find something, in this case, it turned out you find a locus which people, I don't think this shows up well here, but one of the loci shows up with the dichotomous definition better, and the other one with the quantitative definition. And you're not going to really know that beforehand. But the bottom line is, there are lots of ways to kind of be misled. There are lots of ways for misclassification to occur, and these are sort of the kind of canonical categories of misclassification based on whether you're misclassifying essentially at the phenotype level or the genotype level. So you can have people who share the same apparent phenotype, but for reasons that are genetically relatively distinct. And if you lump them together, you will have less power to find potentially either one of those genetic contributors than if you had a homogeneous group. Incomplete penetrance, of course, you can have people who you think are not affected, and so maybe in a case control study you call them controls, but in fact they do carry the risk genotype. And so that will dilute your signal a little bit, perhaps. Variable expressivity, so the phenotype may be expressed in lots of different ways, and if you're not attending to that, again, you might consider somebody as unaffected because they don't have the classic phenotype. And genetic heterogeneity, That is, you may have lots of different kinds of genetic contributions contributing to actually the same phenotype, and that may be difficult in some ways, although less so now that we can just sort of scan the genome and also look at sequence. But there are lots of ways that you can kind of run into trouble. So let's imagine that you are doing a study of Alzheimer's disease. And there is a state of nature that you don't know because you don't know everything. But you do know what you ascertained, what phenotype ended up in your study. And so you did clinical diagnoses and maybe neuroimaging, and you got a bunch of cases, and then you did another clinical screen, you got a bunch of controls. But you can see that some of these are going to be false positives in your case group, and some of these are going to be false negatives. Maybe they just haven't expressed the phenotype yet. The effect of that can be dramatic in some circumstances. And here are some simulations for assuming a sample of, say, 5,000 cases, 5,000 controls for a disease that has about a 1% prevalence. And imagine a locus that has an odds ratio, essentially, for the risk allele of about 1.2, genome-wide significant threshold. And this is what happens to your power based on misdiagnosis rate. And so what you see is that as you increase that, even to, say, 10%, you see a fairly large drop in power. Here are some rules of thumb that people sometimes have used at about a 10% decrease in phenotypic accuracy, maybe about a 20% decrease in power. You may need about 20% increase in sample size there. If your disease is rare, or at least relatively rare, less than 5% prevalence, a false positive, that is calling an unaffected a case, is worse than a false negative. Some things to keep in mind, and actually I'll show you more about why that's true. I think in the interest of time, I'm not going to go into this. This slide just says that in a gene environment study, you've got two reasons to misclassify. You can misclassify the genotype side, or actually three reasons: the genotype side, the environmental side, and the phenotype side. Putting aside the genotype, let's imagine you've done that correctly. If you misspecify either the environment or the outcome, it can have a big effect, but I won't say more about that in the interest of time. Now, what about the controls? How do you define that somebody, again, is not affected, and what's the impact of doing that well or poorly? And you have typically a few choices when people are defining controls in a case control scenario. And partly the way this gets decided is based on practical and cost reasons. So what is a good control? A good control is, what's a good control?
Somebody who doesn't have the disease, absolutely.
Somebody who doesn't have the disease, absolutely. And technically, it should be somebody who was eligible to get the disease, yes. Right? So imagine, this is a little bit of a busy slide. I've got the reference here. It's nice to look at because it gives you a number of different scenarios. But you could have population-based controls. Let's say you're just sampling blood donors or something, and you haven't actually screened them for the disease of interest. People do that very frequently because it's an incredibly easy resource to get in discarded samples and so on, where you've got very large sample sizes, but you don't actually know much about the phenotype. Or B, you've screened them to be non-cases. So you did something and you said, you don't have my disease of interest, which let's say was diabetes. Or C, hypernormal controls. So this is, at the bottom here is the control selection threshold. Imagine there's a sort of dimension of affectedness. So these people are all the way at the tail of having no whiff of anything like your phenotype. That's always going to be more powerful, but it's hard to do that. And you may end up having ascertainment biases when you want to use that control group for something else, because you've gotten a very rarefied group of people. The population-based controls are okay when, and they're about as powerful as actually screening controls, if your disease is relatively uncommon. Because that means that most of your controls will be controls. That is, they will be unaffected. The screened controls are better than the unscreened controls when the phenotype is relatively common. So sometimes we have this dilemma, also, again, a practical one. Do we maximize the sample size or maximize the effect size if we're dealing with complex phenotypes? And so one way to get at this, let's imagine we understand that this phenotype, it's not a perfect definition, it's complicated, there are lots of genetic influences, some of them may be modest, some of them may be large. So one thing we could do is try to pick up subtle signals with volume, volume, volume. Just get a huge sample size. The other is we could try to maximize the effect size if we knew how to do that to define a phenotype where the effect is going to be stronger than perhaps on some big, unrefined, but large category of cases. And the volume, volume, volume approach has paid off. There's no question about it. I mean, genome-wide studies using very large samples that are not necessarily deeply phenotyped has resulted in an enormous advance and number of loci that we now have. It is now possible also to do very large-scale studies with sort of minimal phenotyping using resources that are now available, including population-based registers in Scandinavian countries, for example, which have been enormously helpful. Electronic health record resources that are now becoming ubiquitous. And in fact, at Mass General and the Brigham, there is a biobank that is increasing in size, which has samples linked to the medical record from folks who have consented for very broad kinds of studies. So we now have resources where one can do very large scale, but minimal phenotyping usually. It's possible now to use computer algorithms and natural language processing to mine some of these large-scale resources and actually define phenotypes that are highly predictive. This is actually, these are data from a study that we recently published testing the predictive value of phenotypes derived from computer mining, text mining of large-scale medical records. How do they stack up against in-person intensive phenotyping? And the answer was pretty good. You can train models that do pretty well. So under certain circumstances, that's going to work well. How does it work for genetic studies? We're increasingly seeing cases now where studies that have used those kinds of phenotyping methods have done pretty well in terms of picking up signals that were done in much more detailed, labor-intensive, costly studies. This is work now a few years back from Robert Plenge and others in a study of rheumatoid arthritis risk using electronic health record data and natural language processing and picking up recognized signals. Now, we could try to define the phenotype more strictly, and in psychiatry, there's a long history, as I showed you, of how we've kind of chopped up the phenotypes to be more. This is actually the categories that there were in the 1840 census for neuropsychiatric phenotypes. We now have over 550 different things. And the goal here of subtyping is to sort of reduce the heterogeneity. Can we get more precise? One way to do this is, for example, by stratifying on things like age of onset, which turned out to be very profitable for things like breast cancer, Alzheimer's disease, early successes in identifying risk genes. So when does it help? It helps in part when you know some biological differences that are reasonable ways to stratify your phenotype. Stroke is an example. Stroke as a category is pretty broad, but there are two major kinds of stroke. There are ischemic strokes, which are due to an impairment of blood flow, essentially, and there are hemorrhagic strokes, which are due to bleeding. And among the ischemic strokes, there are three further categories. You can have disease in the large vessels, you can have disease in the small vessels in the brain, you can have emboli that fire off and block blood flow. And if you just did a genome-wide study of stroke as your category, it turns out, wouldn't have nearly done as well as if you respected those subtypes. And here's just some examples from a meta-analysis of VWAS of stroke that was reasonably large and identified 4 loci. All of them were subtype specific. And so all of them were only identifiable by getting down to the level of these subtypes. So this is good timing because Caitlin just walked in. This is great work from Caitlin and Elise Robinson and Mark Daly's group, showing another advantage of subtyping, and that is to say something about genetic architecture. And this, in a way, was an empirical subtyping, that is to say discovering the ways that different genetic contributions act by looking at subfractions of an overall phenotype. So in this case, in a couple of studies, genetic architecture was suggested by stratifying the phenotype, the overall phenotype of autism spectrum disorder, into high and low IQ groups. And it turned out that overall, sort of synthesizing some of this, that low IQ and female sex, which is thought to be, for reasons that I won't go into, is sort of potentially an indicator of stronger genetic burden and more severe symptoms, were enriched essentially for sporadic and de novo kinds of mutations and copy number variants, whereas folks who were more high functioning seemed to have a greater family history, a more heritable, a more, a less de novo form of the disorder in terms of the genetic contributions. And that may help us, this kind of thing may help us parse diagnostic categories and even perhaps lead to new ways of thinking about diagnosis and so on. So there's a case where phenotype can empirically guide us to the genetic architecture to some degree. This was brought up earlier. If you're going to look at a lot of phenotypes, it goes without saying, you've got to correct for the fact that you've looked at a lot of phenotypes. So you need to be specific about what your a priori hypotheses are. You need to be explicit about the number of tests. There are ways to look at phenotypes to reduce data so that you don't have to look at every possible example. But you, of course, have to apply corrections when you do this. But again, you're often faced with this scenario of what do you want to do? You could either do very large scale but more minimal phenotyping, or perhaps you want to fund a study, you're the NIH, of something that's more deep phenotyping, but you just can't get to those numbers. Increasingly, this is not a trade-off, which is a really good thing, because we are increasingly having large-scale phenotypic data resources that actually have deep phenotyping. But one thing that people have proposed is that if you knew something about intermediate phenotypes, that is, phenotypes along the path from genotype to perhaps your more heterogeneous clinical outcome, maybe you would do better in discovering genes, because maybe the genetic architecture might be simpler for something that's more proximal, like an imaging measure or a serum measure of something. Now, people sometimes refer to these as endophenotypes. And again, in the interest of time, I'm not going to go through each of these. But you want something in this vein that is heritable, that is ideally has a simpler mode of inheritance. And I'm showing this. Here's a paper from Chris looking at variants affecting natriuretic peptides that had relatively strong effects. And when you went to the more distal phenotype of hypertension clinically, relatively weaker effects, suggesting that that intermediate phenotype might actually be one where you were picking up a stronger signal. And in theory, having something that captures the genetic effect more closely can have a big effect on power. There's just no question about it. Even a small increase, in this case, of 1.2 genotypic relative risk to 1.3 gets you a lot of power boost. The problem is, what is that better phenotype? And in that case, the fact is that we don't have necessarily a lot of great answers about that. Here's data from a paper last week from Nature, the Enigma consortium, which put together 31,000 brain imaging data sets, essentially, and did a meta-analysis. And these are brain imaging structural phenotypes that are thought to be related to a whole variety of disorders. And the good news is that some very strong signals were identified, for example, in the putamen and to a lesser extent, and also in the hippocampus and so on. These are things that are thought to be underlying many disorders. But again, it took 31,000 to get there. Not necessarily a boost, in that case at least, to your power. But one thing that you may be able to do is use these intermediate phenotypes on the back end. That is, having discovered effects, now you may want to ask, well, how do they play out? And there you're not trying to do discovery. You're trying to do phenotypic characterization. What is the phenotypic spectrum? And that's a whole area that's now available in psychiatry. We're seeing things like this. We recently did this with brain physiology and looking at variants that had been associated with schizophrenia and bipolar disorder. How do those map out onto brain physiologic phenotypes that are related to these disorders that are thought to be part of the underlying biology? So start with established risk variants and now look at what is the spectrum, what is the biology perhaps that's underlying it. The next wave may be VWAS, or at least this is something that is now possible. So again, in electronic health record settings, in registers, in national biobanks and so on, we can begin to look at a vast range of phenotypes. And here's an example from the group at Vanderbilt in which this is smaller than what is now possible, but they looked at a whole variety of SNPs that had previously been associated with a number of phenotypes from the GWAS catalog. And we're able to, using these electronic health records, find most of the signals that had previously been reported, but also find novel associations that hadn't been looked at, in part because people hadn't had this spectrum of phenotypic variation. Okay, so to summarize, when we go through the questions about phenotypes, we want to optimize the phenotype we're looking at, which we can do by trying to have some advanced knowledge of its likelihood of genetic signals through heritability, through GCTA, as we talked about, through other things. Precision matters a lot in terms of power and in terms of our success in identifying genotype-phenotype correlations. And we face some of these challenges. Increasingly, we can have our cake and eat it too, in terms of having deep phenotyping and large sample size. But we often have a trade-off. And I think the bottom line is you're going to get in part what you're looking for or what you're looking at. So however you do your study, you're going to get a scoop of nature. You're either going to get the things that you can pick up with broad minimal phenotyping, or with sufficient power, you're going to get the things that turn up when you have maybe more extensive nuanced phenotyping. Or if you put phenotypes together and do sort of cross disorder work, for example, you're scooping up the things that have those kinds of pleiotropic effects. They're all a part of the underlying genetic architecture, but decisions that you make about the phenotype are going to influence the lens through which you can see any of this stuff. So we have come to the end, and we might have time for maybe one question or so. I want to make sure we get set up for MPG, but thank you very much.